Today I will discuss about hydrogen secretion and bicarbonate reabsorption from the renal tubule. So, sorry. What are the functions of the kidney? We know there are different functions, and one of the most important function is renal control of acid-base balance. The kidneys control acid-base balance by excreting acid or basic urine. Excreting acid urine reduces the amount of acid in extracellular fluid. Excreting basic urine removes base from the extracellular fluid. Renal control of acid-base balance. Each day, the kidneys filter about 4,320 milliequivalent of bicarbonate. This calculation indicates that 180 liter per deciliter filtrate is produced, 24 milliequivalent per liter bicarbonate. Calculation, if it is calculated, it will be this. Under normal conditions, almost all of this reabsorbed from the tubules. That is, bicarbonate that is filtered, almost all are reabsorbed from the tubules. Bicarbonate must react with secreted hydrogen ion to form carbonic acid before it can be reabsorbed. The filtered bicarbonate that filter, it must be reacted with hydrogen ion and will form carbonic acid before it is reabsorbed. So how much hydrogen ion will be secreted? Same to bicarbonate, that is 4,320 milliequivalent hydrogen ion must be secreted. But there is a important thing that additional 80 milliequivalent of hydrogen must be secreted to rid the body of non-volatile acid produced each day for a total 4,400 milliequivalent of hydrogen ion secreted. This extra hydrogen ion must be secreted to rid the body non-volatile acids. In alkalosis, the kidneys usually secrete less hydrogen ion, fail to reabsorb all the filtered bicarbonate, thereby increasing the excretion of bicarbonate. The removal of bicarbonate raises the extracellular fluid hydrogen ion concentration back toward normal. In acidosis, the kidney secrete additional hydrogen ion. Do not excrete bicarbonate ion, reabsorb all the filtered bicarbonate ion, and produce new bicarbonate ion. The action reduces the extracellular fluid hydrogen ion concentration back to normal. Thus, kidneys regulate extracellular fluid hydrogen ion concentration through three fundamental mechanisms: secretion of hydrogen ion, reabsorption of filtered bicarbonate ion, and production of new bicarbonate ion. Secretion of hydrogen ion and reabsorption of bicarbonate by the renal tubules. Hydrogen ion secretion and bicarbonate ion reabsorption occur in all parts of the tubules except the descending and ascending thin limbs of the lobe of Henle. For each bicarbonate ion reabsorbed and hydrogen ion must be secreted. So remember it. Hydrogen secretion and bicarbonate reabsorption occur in all parts of the tubules except descending and ascending thin limb or tubule limb. For each bicarbonate ion reabsorbed, and hydrogen ion must be secreted. About 80 to 90% of the bicarbonate ion reabsorption and hydrogen secretion occur in the proximal tubule. Small amounts of bicarbonate ion flows into the distal tubule and collecting duct. In the thick ascending limb of group only another 10% of the bicarbonate ion is reabsorbed. The remainder of the reabsorption takes place in the distal tubule and collecting duct. This is a figure which indicates where the hydrogen secretion and bicarbonate reabsorption occur. In the proximal tubule, here 85%. Here 85% reabsorption occurs. That is bicarbonate. This is thick part of the ascending limb of the glucose where 10% and rest the collecting tubule and collecting duct. Hydrogen secretion. Secretion of hydrogen ion into the tubular fluid occurs by sodium hydrogen counter transport. Where the sodium hydrogen counter transport present, the epithelial cell of the proximal tubule, thick segment of the ascending limb of glucose early distal tubule. Up to this early distal tubule, proximal tubule, hydrogen secretion occurs by sodium hydrogen counter transport. Remember it. This is a figure where hydrogen secretion occurs in exchange of sodium ion entry. This is secondary counter transport, and this counter transport is maintained by sodium potassium active transport. So if you asked, this hydrogen ion comes from the cell by the action of carbon dioxide with water that produce carbonic acid and forms hydrogen ion, and bicarbonate ion by the enzyme carbonic anhydrase. And from where this carbon dioxide comes? This carbon dioxide comes from the extracellular fluid or from the cell itself by metabolism. This secondary active secretion of hydrogen ion is coupled with the transport of sodium by the sodium hydrogen exchanger protein in the luminal membrane. This is luminal membrane. This is basement. The energy for hydrogen ion secretion against concentration gradient is derived from sodium movement into the cell. That is, sodium enter into the cell, hydrogen ion will get out. This gradient is established by sodium potassium pump lateral membrane. This is I have discussed from carbon dioxide. From carbon dioxide either diffuses into the tubular cell or is formed by metabolism in the tubular epithelial cell. This carbon dioxide acts with water and form carbonic acid and dissociate into bicarbonate ion and hydrogen ion. This hydrogen ion is secreted from the cell into the tubular lumen by sodium hydrogen counter transport. The bicarbonate then moves across the basolateral membrane into the renal interstitial fluid and peritubular capillary fluid. The net result is that every hydrogen ion secreted into the tubular lumen and bicarbonate ion enters the blood. That is, hydrogen ion secreted, bicarbonate ion reabsorbed. We have discussed that to the distal tubule, up to the early distal tubule, sodium ion, hydrogen ion is secreted by sodium hydrogen counter transport. But from the late distal tubule, hydrogen ion is secreted by primary active transport, beginning in the late distal tubule and continuing through the remainder of the tubular system. The tubular epithelial cells secrete hydrogen ion by primary active transport. What are the name of the transporter? Hydrogen transporting ATPase, hydrogen potassium ATPase transporter. The energy required for pumping hydrogen ion is derived from breakdown of ATP. This is hydrogen ATP, hydrogen ion, hydrogen pump. This is hydrogen potassium pump. The hydrogen ion is secreted by consuming ATP. By consuming ATP. Hydrogen ion secretion in these cells is accomplished in two steps. Carbon dioxide combines with water to form carbonic acid. Carbonic acid then dissociates into bicarbonate, which is reabsorbed into the blood, plus hydrogen ion, which is secreted in the tubule by means of the hydrogen ATPase and the hydrogen potassium ATPase transporter. So, at first, hydrogen ion will be produced from carbon dioxide by dissociation of carbonic acid by carbonic anhydrous. Secretion of hydrogen ion in the late distal tubule and collecting tubule accounts for only about 5% of the total hydrogen ion secretion. Next, bicarbonate ion reabsorption. Bicarbonate ion that is filtered by the glomerulus cannot be directly reabsorbed. Bicarbonate ion filtered. In the filtrate, there are huge amount of bicarbonate ion, and this bicarbonate ion must be reabsorbed, almost all of them. But how? This bicarbonate ion is not capable of pass through the luminal membrane. It first, because it is water soluble, and the luminal membrane, that is cell membrane, is water insoluble. So this bicarbonate ion must be transformed into carbon dioxide, and this carbon dioxide will pass through the luminal membrane to the cell. So bicarbonate ion first combines with hydrogen ion that was secreted from carbonic acid and eventually becomes carbon dioxide and water. This carbon dioxide crosses the tubular membrane. The tubular cell. In the tubular cell, this carbon dioxide will combine with water and form bicarbonate ion. This bicarbonate ion dissociates into bicarbonate and hydrogen. Hydrogen ion will be secreted through the tubular lumen, and bicarbonate will reabsorb in the blood. The transport of bicarbonate ion across the basolateral membrane is facilitated by two mechanisms. That is, the bicarbonate ion that was produced from carbon dioxide, it will enter into the blood. At first in the interstitial space by two transporter, sodium bicarbonate cotransport, chloride bicarbonate exchange. Chloride will enter, bicarbonate will get out, and there is another part, that is sodium. With sodium it will enter into the interstitial space. Okay. So, the rest of the like hydrogen ion, excess hydrogen ion that was not um, that was not reabsorbed, that was not reacted with bicarbonate ion, it will react with another two buffer system, phosphate ammonia and ammonia buffer system. For each liter of urine formed, a maximum of only about 0.03 milli equivalent of free hydrogen ion can be excreted. If you form 1 liter urine, the maximum hydrogen ion excretion will be 0.03 milli equivalent. The most important buffers are phosphate buffer and ammonia buffer. Other weak buffer system, there are also urate and citrate buffer. Once all of the bicarbonate has been reabsorbed and it is no longer available to combine with hydrogen ion, any excess hydrogen ion can combine with hydrogen phosphate. Hydrogen ion combines with hydrogen phosphate, monohydrogen phosphate to form dihydrogen phosphate. It can be excreted as sodium salt, sodium dihydrogen phosphate, carrying with it the excess hydrogen ion. Whenever an excess hydrogen ion secreted into the tubular lumen combines with a buffer other than bicarbonate ion, the net effect is the addition of new bicarbonate ion to the blood. In this condition, a new bicarbonate ion is added in the blood. How? By this. That is sodium monohydrogen phosphate, sodium ion. At first, it was disodium monohydrogen phosphate. From it, one sodium um, enters into the cell, and a hydrogen ion combines with this monosodium monohydrogen phosphate and form monosodium dihydrogen phosphate. In this condition, this hydrogen ion, when produced, a bicarbonate ion will enter into the blood. That is formed from the bicarbonate ion. The bicarbonate ion that is formed from water and carbon dioxide. So for each hydrogen ion secreted, a bicarbonate will be produced. Excretion of excess hydrogen ion and generation of new bicarbonate ion by ammonia buffer system. There is another buffer system, ammonia. The second buffer system in the tubular fluid is composed of ammonia and the ammonium ion. Ammonium ion is synthesized from glutamine. This comes mainly from metabolism of amino acids in the liver. This is glutamine. This glutamine comes from amino acid that found in the liver. This glutamine break down into ammonium. This ammonium, this ammonium binds with chloride ion and sodium enters into an exchange of ammonium. Other is ammonia. This also produced from the glutamine. This ammonia directly diffuses from the tubular cell to the lumen, and this ammonia binds with hydrogen ion and forms ammonia, and this ammonium binds with fluorine from ammonium chloride. This hydrogen ion comes from the breakdown of bicarbonate ion by carbonic anhydride that was produced by carbon dioxide and water reaction. So hydrogen is secreted and bicarbonate is reabsorbed that was produced in the cell to the plant. So this is the hydrogen secretion and bicarbonate reabsorption process. That is, it may be mm, discussed or maybe uh, memorized as acid-base balance by the kidney, how kidney maintains acid-base balance in our body. So this hydrogen secretion and bicarbonate absorption is very important for your first year prof exam, and it also very important in future in medicine because acid-base balance maintenance is very important in a patient. If kidney fails, the kidney can't secrete hydrogen and can produce bicarbonate. So the acidosis will develop in our body, and acidosis is very harmful to our body because our body, our blood is always alkaline, and as a homeostatic mechanism, this alkalinity must be maintained. Thank you.